Well, good morning, everyone. I'm anticipating a lively discussion here, so I think we'll go ahead and get started to save that time at the end. Welcome to the February edition of our Spatialomics Interest Group meeting. We are lucky to have one of our NanoString partners, well, a couple of them here today. Maybe he doesn't need any introduction, but I'm gonna read your background anyway, just for everyone's sake. So we have Jerid Robinson here today. He's a field application scientist for NanoString, and I think your title has changed, but I can let you let us know about that. But he's a FAS for both the DSP and the CosMx platform. Jerid earned his Ph.D. in pharmacology from the University of Minnesota and completed his postdoctoral training at Emory University, where he focused on how PTH activates intracellular signaling cascades in T cells and how those signals eventually led to increased bone. Jerid is an experienced immunologist with a background in pharmacological therapies for osteoimmunological disease. Jerid, we're very excited to have you here today, and I will just turn it over to you. Thanks for coming.
Yeah, sure thing. All right, thanks, Grant. I really appreciate it. It's been a great working relationship with you and the team. You and Fernando have just been fantastic. Anytime that we have something come up or something new, you guys are always really open to us presenting, and then also just really great discussions. So I really appreciate it. But yeah, as Grant mentioned, I'm Jerid. I did go to the University of Minnesota in the Department of Pharmacology. So some of you may know me from those days. I want to make sure you can see my screen. Is that accurate?
Yes, we got it.
All right, fantastic. That's what we're looking for. Okay, so I'll turn my video off just to sort of save on bandwidth. We have no idea what's happening with the snow, but yeah, I live in Minneapolis, down in South Minneapolis. I manage the West FAS team, so there's 8 of us in West. West does not mean the Mississippi River, it actually means everything west of Pennsylvania. So quite a big territory, but yep, my home base is here, and I happen to be home this week, so it's really nice. I'm going to talk a little bit today, sort of an overview of GeoMx. I'm sure most of you have used the platform. If you haven't, please feel free to reach out to me. We can chat about it, how we can implement it into your projects and your research goals. But yeah, we're going to do a little bit of contrast comparison, a little bit about GeoMx, a little more of a deep dive into the CosMx system that we've had out for a couple months now, and that's our spatial molecular imager. We'll often refer to these interchangeably as GeoMx and DSP, or CosMx and SMI. So forgive me if I sort of use those interchangeably. All right, so let's just go ahead and get started. Make sure I've got my laser pointer up. There we go, perfect. Okay, so we show the slide. I can't tell you how many times I've shown this slide. We really do show this progression from doing bulk gene expression analysis, things like qPCR, or doing a bulk gene expression using RNA-seq, or as I like to have people do, use our nCounter technology. It's a mid-level sort of mid-plex gene expression platform. But over time, we've moved on into some other realms. 10x brought out their Chromium system a number of years ago now. It's crazy to think that was a decade ago, doing single-cell genomics. And so we learned to take something that's quite blended. We can't really separate cells out, to having the ability to separate cells and do single-cell RNA-seq. From that, though, we really want to understand how are these cells related, and then how is their proximity to one another, their spatial information, how is that affecting gene expression? And so we've moved on from that into more high-plex spatial biology. There's a lot of systems out there. I won't say that we're the only game in town. I do think we're the best game, but we're not the only ones. We have 2 platforms though, and so I'm sure many of you are used to this GeoMx platform, our digital spatial profiler. We've had it out since the end of 2019, and you have the ability to do multicellular. We're talking about cell neighborhoods, cell populations, down to around 100 micron diameter circles. You can go a little bit lower. And the benefit here is the ability to do the entire transcriptome. We have whole transcriptomic analysis for human and mouse. We're also coming out with a canine immuno-oncology panel pretty soon. With the CosMx system, this is something that just came out. We shipped our first commercial unit, our non-beta units, at the end of last year. And so with this one, we have the ability to go down to single cell and even subcellular localization of both genes and proteins. The difference here is instead of doing the whole transcriptome, currently we offer up to 1,000 plex RNA and a 64 plex protein. And so a little bit of difference there just in terms of plex. And then obviously quite a bit of difference between the resolution. So give a little bit of background on how the GeoMx platform actually works, just for those of you who maybe aren't experienced using the GeoMx platform. You stain with both morphology markers. So these are going to be, you can think of it very much like a digital H&E or a fluorescent H&E stain. They're used only to identify regions of interest. So it's just a way to sort of map your tissue to figure out where you want to do some additional profiling. And then those are going to be fluor conjugated primary antibodies for the most part. In addition to that, we add in probes. And so those can be barcoded antibodies that have a photocleavable linker, or they can be in situ hybridization or ISH probes if you're looking at RNA, and that additionally has a photocleavable linker and a DSP barcode. So once you select your regions of interest based off of your fluorescence, you then instruct the instrument to UV photocleave these oligos, you collect them, and then you process those, and you can count them either using nCounter if it's below 96 targets, so a lower plex readout, or if you're doing up to the transcriptomic level, then you would utilize our NGS readout. In terms of the optics, they're pretty cool because you have the ability to do things like choose geometric shapes, do contouring, or you can do concentric circles, or you can take a completely discovery-based gridded approach. Within these regions of interest, you can do a secondary thing, which we're really the only ones that can do this, and that's these areas of illumination. You're able to further subdivide these regions of interest based off of your fluorescent signal. And so the classic example that we use all the time is looking at this stain here. So we have pancytokeratin positive cells that are going to be colored by yellow. The nuclear stain here is in magenta, and we can use our digital micromirror array in the system, around 2 million 1 micron squared microchips that can be instructed independently to either direct UV light into the tissue, thereby photocleaving those oligos, or directing light away from the tissue, thereby not focusing on that. And so we can profile 2 separate cell populations within a given ROI. And I think the upper limit is 7 because we have 3 colors plus a nuclear stain. And so you can do potentially 7 subdivisions of an ROI and then collect those areas of illumination separately, and they're profiled separately while still maintaining the spatial information. So it's really cool to kind of understand what may be changing in a microenvironment to different cell populations. I always like to talk about this just because we've seen a number of times setting up a GeoMx experiment isn't difficult, but there are steps that you need to make sure that you take care of. And the first is going to be tissue. We can work with a number of different tissue types, and so I won't really belabor the point, but if you have tissue, we likely can work with it. We work well with FFPE, which is different than some other competing technologies. We work quite well with tissue microarrays and fresh frozen tissue. The way that the GeoMx works, though, we want to make sure that we cut those blocks and put them onto slides within a couple of weeks of running them for RNA, just because we want to avoid the risk of having your RNA undergo hydrolysis. That can lead to lower signal, so we want to make sure that we do that. With fresh frozen tissue, you can use that as well. Still going to want to cut those within 2 weeks of running them. But with fresh frozen tissue, you can use OCT buffer using a cryostat. With FFPE, obviously, it's going to be formalin fixed, paraffin embedded. We prefer for both of them that we have the section thickness between 4 and 8 microns. 5 is pretty typical for most histology groups. And then for the fresh frozen tissue, we do understand that you might end up having some issues there with getting 4 to 8 microns. And so we've seen some data going up to 10 microns in thickness for fresh frozen. It's a bit more difficult to section at times. We recommend keeping those slides at 4 degrees Celsius with a desiccant until you're going to run them. This just keeps the risk of hydrolysis down. If you have fresh frozen slides, we recommend keeping those in the freezer. With protein, it's a little bit more permissive in terms of how long after putting onto a slide. In terms of the slide, you don't have to have anything in particular. They just need to be positively charged. So a typical Fisher Superfrost Plus slide works quite well. And then this green area here is going to be your scannable area. And this is just a demo showing mouse brain. You can fit 6. I actually have a group in Michigan that has collected 9, and I'm not sure. Maybe they grew up with a family member who did UPS packing or something, because yeah, they can get 9 brains on. Is wild. Now, in terms of morphology markers on the GeoMx system are going to be limited to 4 colors. One we prefer be occupied by the nuclear marker. In case of the things that we sell sort of off the shelf, that's going to be utilizing Cyto 13. Since we use UV light to photocleave those oligos, you can't use DAPI. And so as a result, one of these 4 channels we prefer to be occupied by a nuclear stain. The other 3 can be utilized for, again, your morphology markers, those fluor conjugated primary antibodies. And so this just sort of shows that. I like to use Alexafluor nomenclature. It's just a bit more specific. And so the channels that we prefer are going to be Alexafluor 48, 532, 594, and 647. We can sort of talk about these profiling modalities. I really won't belabor the point here. We have some white papers and tech notes on these. So if you have any questions, feel free to reach out and I can send you that information. We do have some minimum and maximums, though, that are sort of important to understand before you get into your experiment. The minimum size that you can choose is 5 microns by 5 microns. I wouldn't recommend that, just primarily because you're not going to get a lot of material to profile. The maximum size is going to be 660 by 785 if you're doing a rectangle. If you're doing a circle, it's going to be a circle with a diameter of 660 microns. For the protein assay, we recommend having somewhere in the neighborhood of 20 cells. You can certainly use fewer. You'll have some low expressors drop out, so there are caveats there. And then for the RNA assay, I think a lot of our documentation will say we need 200 cells or between 100 and 200. In the field, I've seen that if you have 50 cells and around 1,000 square microns of area, you're usually okay. And so fewer, obviously, than 50 can be used with caveats. We even have groups that do single cell analyses, and so it is possible. I know you had Miranda Orr on recently. She does single cell all the time on the GeoMx, and so it is possible, again, with some caveats. And just remember that these numbers are based on not just the ROI, but if you further subdivide into segments or areas of illumination, these cell numbers refer to a collection. And so be it the entire ROI or the collection of a segment. And then before I really get into more of the CosMx, because that's hopefully why you're here, I do want to talk about some of the data analysis options. I know that there is a strong bioinformatics group there at the University of Minnesota. And so, we have the ability to run things on the instrument. And so we have our GeoMx DSP analysis suite that's going to be on instrument. You have the ability to remote into the instrument, especially for things like ROI selection. You don't necessarily have to be in front of the machine for that. And then you can also do the data analysis here. A lot of times the core is going to do some of the QC checks perhaps, and then from there you can decide whether you want to do on instrument or if you want to proceed into doing something a little bit different in terms of your analysis. But we'll show you some of the visualizations. You can do volcano plots, you can do heat maps. This is going to be unsupervised clustering analyses. And then you can also do pathways using our gene set enrichment analysis. We utilize R for that, and so the R pathway IDs are incorporated into this visualization. If you didn't want to do anything on instrument, though, you want to do everything completely off instrument, we have GeoMx Tools. And so GeoMx Tools is coded in R. We have a number of different resources here that you can utilize. And it's maintained by one of our own, Nicole Ortiguero. And so that is an option, and you can take this, it's open source, you can modify it however you want, and we have a number of people, especially that have really strong and large bioinformatics groups, that take this as sort of a template, and then they create their own pipeline. So that's an option as well. Then finally, if we have time towards the end, I would like to show you what CosMx looks like. I have access to it, and so I can just sort of show you what the visuals look like, and then also show you what data analysis would look like on the CosMx platform using CosMx data. Looks like we got a couple of questions. Oh, sweet. Thanks, Grant. Just want to make sure I didn't miss anything. All right. And so any questions about the GeoMx? I know I kind of ran through that quickly, but my understanding is that this group has quite a bit of knowledge about how to use the GeoMx system. So I really wanted to focus more on CosMx. All right, sounds good. Okay, so moving right along into our CosMx spatial molecular imager. So as I mentioned, we just released our first commercial unit at the end of last year. And so, we're going from things like this, and as I sort of mentioned, looking at that smoothie fruit salad, fruit tart visual, we're going from understanding how cells maybe cluster and what their visual associations are. Utilizing a UMAP, you can find different populations of cells using single cell RNA-seq. It works really well, right? I mean, I'm sure many of you have published data showing a UMAP just like this. But what we want to map the really awesome power of single cell RNA-seq to spatial interpretation. So not only do we want to know what cells are where and how they're related, we want to know where they are and what they're doing and why these cells maybe are a little bit different and show up in a different place on the UMAP compared to other cells. Why? Because there's some tissue architecture here. There's actual spatial information here that we need to get. And so this just sort of shows a little map, 1 centimeter by 1 centimeter, looking at both our cell typing and our cell-to-cell interactions. And this is what the CosMx box looks like. It's about the same size as the GeoMx, a little bit deeper, but about the same size. And so again, we can do, in this particular example, we're looking at a whole tissue section. We can do just over 1.4 million cells in this particular lymph node. This is FF. We collected a number of fields of view. You can sort of see them highlighted by these little boxes here. These are FOVs. We focus in, we get single cell resolution. We can also do subcellular resolution. The resolution is around 50 nanometers in the XY plane, but we can get down to a solid micron when we're looking at it. So I always like to start with things that things that you're familiar with, and maybe something that's a bit new. And so when we're comparing GeoMx to CosMx, if you recall, we talked about the profiling reagents that we can use. We have protein reagents, so we have fluoroconjugated, or I'm sorry, we have an antibody with a UV photocleavable linker and a barcode. And so the readout really is this DSP barcode. Most often we'll sequence that, but like I said, you can use nCounter for readout. Same thing with the RNA. We have a complementary sequence here. This is just an in situ hybridization probe, UV photocleavable linker. And again, the readout is going to be this DSP barcode. With CosMx, things have changed a bit, and so we use a technology that we call hide and seek, which is something that we've had around for a little while, and we were just trying to figure out how best to implement it into a technology. And so there are times when you have really great ideas, and you're just waiting for the technology to catch up. That's kind of where we were a few years ago before CosMx came out. And so we have this target binding domain. It's very similar to the target binding domain for our ISH probes on GeoMx. Same length, they're between 35 and 50 nucleotides in length. And then attached to that, rather than a UV photocleavable linker and a barcode, we actually have a readout domain. And so that's the first difference between GeoMx and CosMx, is on CosMx, you're going to be able to do your readout on instrument. You don't have to run your slides and then take some 96-well plate and then process that. All of the readout is going to be on instrument. And so this readout domain has a number of different binding sites. You'll see 4, and we'll go through that in just a moment. But essentially, attached to that is going to be photocleavable sites. So we're going to cleave it off when we're done reading that signal. And then we have a really boosted reporter signal. So you can see this, this lattice work here. And we'll go into that as well. Similarly, we also have an antibody with a linker and then the readout domain. So kind of similar architecture. It's just that we're able to do the readout on instrument versus cleaving the barcode and counting it later. And so the workflow itself is going to be really similar. I'll actually show you in a bit what the workflow actually looks like and how that would compare to GeoMx, but it's really similar. And so, standard lab things that you already have for GeoMx, you really don't need much else for CosMx. I think the only addition is going to be a sonicator, which most labs have anyway. So we'll start with, FFPE tissue. We can also work with fresh frozen. We'll go through a permeabilization and fixation step. We'll do an antigen retrieval and target retrieval. Then we'll do an overnight hybridization. This is going to be really familiar to those of you who have done a GeoMx workflow with RNA. We do an overnight hybridization step. The next day you come in, and then we set up the flow cell. And so that is a little bit different than what you're going to see working with GeoMx, this flow cell assembly. And that's because we're going to be washing our probes on, photocleaving them, and then washing them off. So we need a flow cell set up just so that we can sort of continuously run liquid through the slide. So a bit of nomenclature. You've got your target, that's in gray. We have our in situ hybridization probe. It's not fluorescent, like I mentioned, somewhere between 35 and 50 nucleotides in length. We go ahead and add on that reporter probe. And so this is how the probes look when we hybridize them overnight. You've got this readout domain, and there's 4 landing domains for the reporter probe. And so that's going to be after we get to the end here. And we can talk about the reporter domain. And then let's actually show what it looks like. So if we run through, you see we add in our in situ probe. This reporter probe is fluorescent, and it contains 4 reporter landing domains here. This is the reporter readout domain. There's 4 different landing domains. And you go, well, Jerid, if there's only 4 landing domains, how do you get up to 1,000 plex? Well, the way that we do that is if we take our particular target, we've got our in situ hybridization probe, we've got our landing domains for our reporter probe, and then we have up to 4 colors. And so since we have these branched readout probes, they're quite bright. We have 4 different dye colors. So we have a photo channel, a 4-channel readout. And the way that we do this and the way that we proceed through this workflow, and you'll understand when I show you this table here in a bit, the time that it takes to do this. We do 15 minutes with the in situ with the reporters. We wash away any unbound reporters, we image, and then we remove. And so the removing of the reporters is going to be really similar to what we've seen before, and that's going to be through a UV cleavage step. And so let's just sort of walk through this. We'll get through some of this stuff here. We won't really talk about it too much. The nomenclature is important, but I'll describe it as we go through it. I'm happy to share these slides when we're done. And so the way that we do this is we have on and off events. And so for our 100 plex RNA barcoding kit, we have a 4 on, 4 off. So it's going to be 8-bit. When we go to our 1000 plex, we go up to 16 bits. So we'll have a scenario where we'll have 4 ons and 12 offs. And so the way that this works is we map these barcodes in X, Y, and Z positions, and then each gene has a series of barcodes. And so in counter technology, we have 4 colors and 6 spots. In this particular example, we have 4 colors and an off. And so this value of 1 is going to mean that there is a color, and then we have to identify which one it is. And then we have a series of offs. And so if we have gene 1 and gene 2, we look at these readout domains, we can see that they're quite different. We have red for the first round, green for the second round. They're off until we get to the 7th round, in which we have a yellow, off again until we get to round 16, and that's yellow again. And so if we run through this, we flow on. We give them 15 minutes to hybridize. The only one that's hybridizing in this first round is red 1, because there isn't a 1 here. We then look, we've got a red 1. That's for the first cycle. We photocleave, wash it off. We flow the reporters on. For number 2, we have a green here and a yellow here. And then we photocleave, remove, and then we go through this 16 cycles to get to our readout. And so, like I said, depending on whether we're doing a 100 plex or 1000 plex, it's 8 or 16 cycles. And then we do this twice so that we ensure that we can account for errors. Because again, you want to make sure that what you're seeing is true signal, and you want to make sure that the values match. And so what we offer in terms of our RNA assays, we have currently a human universal cell characterization panel. That's 1000 plex RNA. We also have a more curated one that's our human immuno-oncology panel. That's 100 plex. We have a mouse neuro 1000 plex, and then finally we have our human IO 64 plex protein panel. Now, in addition to these predefined panels, you can add on some spike-ins. So we have our ability to add between 7 and 50 user-defined genes in the 1000 plex, 7 to 10 in the 100 plex RNA panel. We also have the ability to do a de novo custom panel. And so that allows you to look for up to 300 targets, and then you can add a spike in on top of the standalone de novo custom panel. And so just looking at the really clear differences, if we start with just where the tissue is going on the slide, right? So with CosMx, again, we can do subcellular resolution. With GeoMx, we have a much higher plex. Readout is on the instrument on the CosMx because of the fluorescence. And then we obviously have an external readout for the GeoMx. If we look at the slide, though, this is, again, just a typical, positively charged slide. Superfrost Plus works quite well. If you have tissue that you're afraid is going to fall off, then you can certainly use a like Apex adhesive slide. But the area, the scannable area is a bit different. And so here we have a 20 by 15 scannable area. Here we have a 35 by 14, so a little bit bigger. The reason that we have to have a smaller area here is because we have to apply the flow cell onto it. And so you can see the flow cell outline is here with the inlet and outlet ports. We also have some differences when it comes to our morphology markers. CosMx, we actually call these segmentation markers. And the main reason is because we're going to utilize these not just to identify interesting cell types. The most common one is utilizing pancytokeratin and CD45. We also utilize this to identify the nucleus and then also to identify the outline of the cell. And so we have a membrane or cell boundary floor. So the way that these are set up, they're a little bit different as well from the fluor conjugated primary antibodies that we use. And so the purposes that they serve are really for FOV selection. So you can identify regions of interest and lay down a field of view. I'll show you what the FOVs look like. They're a little bit different than what you see when you choose ROIs. And so it's important to kind of go through that. These guys are not conjugated directly to a fluor. They actually have a very similar readout domain to what you would see on the targets that we're looking for. The only difference is that it's a very short readout domain. So you have a universal handle, and then you just have one site. So one reporter landing spot versus 4 in the readout domains of the targets. And then again, that universal handle is just for purification of the conjugate. We have the ability to use DAPI. Since we're not using UV light to photocleave oligos, we actually use UV light for DAPI. And then we also use UV light to cleave off those reporters that we then wash away. And so we can utilize DAPI for the nuclear stain, and then we have a cell membrane marker utilizing beta-2 microglobulin. Those are required because that's how we can do the cell boundaries and the cell assignments. And so the kits that we currently offer are going to, again, fall into these 5 channels. You'll recognize these number 2 through 5. These channels are ones that you've already seen on the GeoMx. And then this SMI channel 1 is just the same UV channel that we utilize in the GeoMx. We just have the ability now to utilize it for a DAPI stain. So we have a nuclear segmentation channel, our cell boundary channel, and then you have 3 channels that you can use for whatever you'd like. And we can kind of go through that. At the moment, what we have is pancytokeratin and CD45. Those are going to be our supplemental segmentation kits. Again, these are going to be mainly for field of view selection, and if you wanted to approach any sort of segmentation. These segmentation kits are going to be pretty set, DAPI again, and then this beta-2 microglobulin in the Alexaflor 48 channel. If we just take a look, so this again is that flow cell. This is our 20 by 15. We're looking in here. This is what a field of view looks like. These are going to be fixed, so they're not adjustable like you've seen before with the GeoMx platform. And so you can choose just individual fields of view. You can just lay one down here, lay one up here, lay them wherever you want. And the idea is that you can take that approach where you put down specific fields of view in specific places based off of your segment markers. Or you can do a gridded approach, so you can lay down, up to 1,000 fields of view, that full 300 square millimeters. So, you can do something quite small or something quite large. And so the turnaround time on the instrument, since we utilize the instrument itself for readout, turnaround time is going to be determined by the plex. So whether you're doing 100 or 1,000. How many flow cells, so that's how many slides you have on the instrument, and also the number of fields of view per slide. And I have a handy little turnaround time visual here. So if you're doing 100-plex, we have it split into 2-slide and 4-slide. 1,000-plex, same thing, 2 and 4, and then the protein. And then this shows you if you're choosing 62, 193, 386, 772, or just the whole darn thing, and what the turnaround time is. What we validated so far is the 100-plex 2-slide goes out to about 11 days. The 1,000-plex 4-slide here, if you do the entire FOV, we can chat if you really want to do that. it takes a bit, and so we can certainly talk about that. And so how do we actually do this segmentation thing, right? How do we actually define the cells? And so the way that we do it is we take a Z stack, multi-channel tissue images, we combine that together and we do some processing. So we have image pre-processing. We try and make sure that we have good autofocusing on the nuclear stain. And so that's how we generate this nuclear model output. Then utilizing this Z stack, we put together our cell membrane model. And then finally, we can identify the specific cell boundaries. So this is what it looks like. This just shows you inputs for our cell segmentation. This is looking at normal liver. So we have DAPI, we have cytokeratin, we have CD45, and it just shows you the channels and then how it comes together into this segmentation that we have. And I'll show you what this looks like on the atomic system in just a second. And so I want to give these last few slides before we switch over and look at the atomics platform, just to show you kind of the similarities and differences in the workflow. And so we have an overnight baking step that's a little bit different than GeoMx, where we have a shorter baking step on the day that you're starting your slide prep. We have a very similar deparaffинization and rehydration step. And then target retrieval is going to happen in our pressure cooker, which I know Grant has one. I've seen it. Going from there, there is a slide drying step that's a little bit different than what you would do in the GeoMx. But then we go straight into setting up our protease digestion. And so that's quite a bit different. We don't really have a digestion step in the hybridization chamber. We have preparation for our fiducials. And so I won't talk too much about what the fiducials are specifically, but what they do is they're a way to map the slide. And so the fiducials are essentially a little fluorescent grid that ends up going onto the slide. It sticks, and then it allows us to have reference points so that we can make sure that all of the images that we take are properly aligned. Once we do that, we do a postfix step just to make sure that all of the target retrieval that we did and then laying down the fiducials gets stuck in place. And then we set up our overnight probes. And so this utilizes a slightly different cover slip than those hybrid slip covers you're probably used to using, but the application is very similar. So it's just an overnight in situ hybridization using those probes. You come in the next day. This is going to be very similar. You do stringent washes to remove off-target probes. Then you do your nuclear and morphology staining. This will be using DAPI instead of those cyto dyes. You add in this NHS acetate. You assemble the flow cell. I'll show you what the flow cell assembly looks like, and then put it onto the instrument. So when you're thinking about choosing a spatial platform, there's a lot of things that you want to consider. The first being Plex. And so when we think about Plex, right now we have a commercially available 1,000 Plex universal human cell characterization panel, and then that mouse neuro panel. Those are both 1,000 Plex. For GeoMx, we have transcriptomic level analyses for human and mouse. So 18,000 for human, just over 22,000 for mouse. So it really depends on the Plex that you want to do. Now, we just released, I don't know, for those of you who are at AGBT down in Florida, shame on you for leaving us up here in the cold, but I hope you had a good time. At AGBT, our Chief Scientific Officer, Dr. Joe Beecham, he actually demonstrated that coming out next year, we're going to have a 6,000 Plex RNA panel. And so we've already shown that. We have data on it. Feel free to reach out to me if you want to take a look. But it's still not quite transcriptomic level, right? So 6,000 is not 18,000. And so that's something you want to think about. Panels. Right now we have the ability to, again, kind of a limited panel for CosMx. But if you look kind of into the genes that are available and the proteins that are available, 64-plex, 1000-plex, there's a lot there. However, if you want to do the entire transcriptome, then you would want to go the route of GeoMx. If you want to do something custom, it's probably a little bit easier to get that through GeoMx as well right now. And so just things to think about. When we're talking about resolution, obviously that's a big one. Subcellular resolution on the CosMx, sort of neighborhood level resolution when we're talking about GeoMx. And then throughput. Do that table, don't want to scare anybody, but CosMx is going to take a little bit longer. And that's primarily because we have the analysis and the readout is on instrument. And so as a result, throughput is going to be a little bit less throughput on the CosMx than you would on the GeoMx. And so that's also a consideration. How long is it going to take you to get your data? And then this is just, this is sort of the meat and potatoes thing is, what is your biological question? I've had customers that I've had to turn away, sorry, Elliot, that I've had to turn away because their biological question didn't have a spatial component. And it's one of those scenarios of, well, you might encounter platform because you're not asking a biological question that actually has a spatial aspect. And so when you're asking yourself, what do you want to get out of this? Are you interested in cell-to-cell interactions, or are you interested in understanding the interplay between populations? Do you need to understand target localization inside a cell, or are you more focused on understanding how groups of cells are going to react to a certain disease or drug or stimulus or treatment? And so those things are really what's going to separate out, doing a GeoMx run versus a CosMx run. We find that they're quite complementary, and it really does depend on your biological question. We have a number of people, and I'm sure you've seen them talk in various marketing things. Grant Kohler is a huge example. I've worked with him quite a bit down at St. Louis University, and he does both because there is value in doing both platforms. It really does depend on your biological question. And so I kind of want to end here, and I'll leave a couple minutes for questions before we hop over to look at the AtoMx platform. But yeah, we have the ability, GeoMx is going to be linked up to the AtoMx Spatial Informatics portal. CosMx is, you have to have AtoMx for CosMx to run. And so as a result, I kind of want to show you what this platform actually looks like, because I don't know if we've shared it very much with customers. And so I'd love to show you that. But I'll pause for just a moment for any questions that you might have. I didn't see anything in the chat, but I'm happy to answer anything offline. I can put in the chat my particular email address. It's very easy. It's just jrobinson@nanostring.com. And I'm happy to set up, ROI, selection demos, data analysis demos on the GeoMx, just really whatever you need from a technical standpoint to better understand the platform. All right, so let's hop over and take a look at our AtoMx platform. That's always kind of cool to be able to see because I don't know how many people have actually seen AtoMx in the wild. So let's pull that up. Don't worry, I'm still here. I'm just going to resume my share. There we go. I had to pull up this demo here. So when we log in, this is the AtoMx Spatial Informatics Platform, or SIP. You have the ability to link not just your CosMx, but also your GeoMx. And so we color-coded them just to keep sort of the confusion down. Extracellular vesicles. Oh yeah, okay. So you'll have to, let's see, one of the platforms for extracellular vesicles, RNA-seq, or protein expression on the surface of EVs. We've had a couple of groups do that. It's a little complicated, right? Because you're working with with EVs and not actual cells. But yeah, we have done that in the past. That's probably something that's going to require a bit more in-depth conversation. But yes, there is a way to do that. So it's a qualified yes. But yeah, we can talk about it. So yeah, you have the ability to put your GeoMx as well as your CosMx onto your AtoMx platform, and they're color coded just for ease of use so that you can understand studies that were created either on a CosMx instrument or GeoMx. So if we go up here to home, this is what it looks like sort of from a gallery standpoint. We have studies that are created, and so these are going to be data analysis studies that have been set up. GeoMx green, CosMx in blue, and so you can see that they're all kind of together. Now you can put them into folders, And so the folders that you've seen on the GeoMx platform are going to be called collections here. And so you can set that up by lab, or you can set it up by project, or you can set up collections really however you want. And you have the ability to view your content, team content, or shared content. So you can really have the ability to lock down who's seeing your data, even if you're under the umbrella of the core facility in terms of the atomics. Now, if we go in and look at this tonsil study here, oh, and one last thing, flow cells and scans. So we refer to the scans that we're looking at on CosMx as flow cells, and then the scans are going to be those on the GeoMx. And so you can see all the ones that are showing up down here are going to be from GeoMx. But let's take a look at this tonsil study. And so if we look here, we'll open this up, and this shows tonsil run. And we only chose 4 fields of view. That's typical for when we're doing a training run. And this shows our pipeline structure. So this is similar to what you've seen working with GeoMx, where you start with your initial data set, go through some QC normalization steps, and then you can get down to looking at principal component analyses, and then also UMAPs. And so if we just take a look at the UMAP, we can make this big. And this is just showing a UMAP for our cell typing. And right now we have them separated into A through L in terms of the cell type. You can certainly select specific cell types to look at within a population. You can also change things a bit as well. Right now we just have them listed as A through L, which is not particularly informative, but as we come out with with this Atomics platform version 1.1, this will be a little bit different. This is just for demonstration purposes. So you can focus in on this. You can look at normalization. You can look at saturation. You can also change the settings a little bit if you want, and then save it. This is pretty standard. If we go back though, you can also look not just at the FOVs that were shown up here. We just have one checked, but you can actually look at all of them together, or specific ones. It's really up to you. We also have the ability to do cell typing, so that's an option as well. And then there's a few other things that you can do, but I know that we're probably getting close to time. So I just want to show what it actually looks like in terms of the flow cell, so that scanned image. And so you can see we've got just a normal tonsil here. We utilized DNA, so that's going to be our DAPI stain, pancytokeratin. In this case, we're targeting macrophages, so we're looking at CD68. CD45 is a broad immune marker, and then we use that beta-2 microglobulin for our membrane stain. So if we zoom in, you can select these regions. You can also go in here and look at the FOVs. It'll tell you how many cells there are, and then it'll also give you the coordinates. And so we can focus in just on FOV number 1 to kind of start. We can also get rid of this little mini map because it's just in the way. We can also get rid of this legend just so we can sort of free up a little bit of space here to look. So if we look at FOV number 1, we'll zoom in, and you can see that our cell segmentation algorithm is already at work. We can see the nuclear markers in blue, and we don't really see too much of the membrane marker. We would if we alter the settings, and so you can do that as well. Going into our render settings here, you can, just like you would in the GeoMx system, you can modify how bright or dim they are, and then you can also utilize, since these are pseudo-colored, you can color map them, again, very similar to what you see with the GeoMx platform to whatever you want. And so these just show you all the channels. This is red, yellow, green, blue, and UV. And so just taking a look, you can see how many cells you have, and then you can look at specific gene expression profiles as well. That would be on the analysis side. But yeah, you get some decent little images. You can look at them sort of this way, down at the cellular level. There's not a lot there in that particular FOV, but we can go over to number 4, where we can see some different things here. Zoom in, and then you can start to see some of the associations with those segmentation markers. So again, we're not looking at gene expression here, we're just looking at those fluorescent tags that we have. I'm bringing the legend back up just so you can sort of see what I'm talking about. We have pancytokeratin. We've got a pretty strong membrane staining here. Again, you can modify that to make it brighter or dimmer, and just sort of play around with it. Again, this is just a demo data set. This is what we typically use for our training. And so, yeah, just gives you an idea of what it looks like. Any questions about this? Oh, I should also mention, we do have the ability to do overlays just like you would in the GeoMx system. So you could do an overlay of, say, an H&E stain, for example. So any questions? I do want to leave time for any questions. So I'll turn my video on just so you can see I'm a human. Hi there. Haven't died yet. I've already been shoveling quite a bit today. So yeah, any questions though about how the system works, or questions about sort of resolution or tissue, or really any technical question you might have about either the GeoMx or the CosMx system? I'm happy to help.
Jerid, I anticipate some questions, and if you don't mind, I do want to jump in real quick
Yeah,
ahead of
of
time.
course.
I just, for this particular group as well, we have people coming going, make it very clear. We do have a CosMx coming. So we are happy to start talking with you, our researchers, get you in touch with Jerid to start planning these things. Maybe I'll ask you, do we know when it's coming?
Let me see. Actually, I can take a look. I'm not going to make any promises, but yeah, we have a smart sheet where we talk about it. I don't actually know when it's slated. Maybe we'll take that offline. But yeah, we have a running list of what's been installed, what's been delivered. So yeah, I can take a look and see.
No, thank you. Figured I'd put you on the spot in front of
This
everyone.
year, definitely this year.
No, and before we open it up to more questions, I do want to just have one. I appreciate the walkthrough on the chemistry. There are
Yeah.
kind of generally several of these now that are doing this in situ sequencing. So, you know, we know you can build a nice instrument, and I think a lot of these will come down to the quality of chemistry and how that works. So, I'm intrigued by the photocleavable. I like that. Do you have any comments maybe about the hide and seek, what it's called, and maybe how it compares to other platforms or technologies, like something like Mirrorfish, for example?
Yeah, that's the classic one. So yeah, I don't feel bad about having us come up against you know, Mirrorfish and Merscope. Yeah. So in terms of how we approach it, obviously Plex is going to be coming into play, and then time. And so for our system, we seem to have pretty solid resolution. That doesn't seem to be an issue. Plex is just a matter of time before we add more. I mean, we started with 100, we went to 1,000, and you know, now we're talking about 6,000. The technology itself and the fact that we're using those really bright floors, I think there's a lot of commonalities now between the different groups. We're lucky enough to where we've identified and sort of modified our existing approach from GeoMx to have the ability to have those readout groups. So it makes it really specific. It's super specific, it's super bright. And then we run 2 rounds of that so that we ensure that the signal that we got is really where it's supposed to be. And so I think at the moment we're solid on resolution. We have a fantastic platform for visualizing and analyzing data. Yeah, it puts us in a good spot. But yeah, like you mentioned, there's a lot of players out there, and it really is. It's always one of those things. We get into these spec wars, and I go, nobody cares. I mean, to some degree, they do. But I take my iPhone, for example. I have friends that talk about, oh, I've got 20 gigs of RAM on my Android phone, and I go, mine just works. And so I think at the end of the day, what people want and what we've sort of gotten feedback from and incorporated into the CosMx system is ease of use. You just want to put it in, you want to choose your regions, and then hit go and have it work. And that's really where we're kind of hanging our hat, is the ability for things to just be plug and play, for the analysis to be seamless, and for you to get data. Because in this really competitive, you know, world of spatial, you've got to be the first. So yeah, that's
Awesome.
That's sort of the Cliff Notes version.
No, thank you. Appreciate the discussion. We're very excited about it, and I'd be remiss if I didn't thank Laura and Dave Bernor for helping us achieve that and getting that here. So thank you to them as well. And I'll go ahead. I see Andy's on screen, and I'll open it up for other questions as well.
Sure.
Oh yeah. Hey, Jerid, Andy, Nelson. So one of the quick questions I have is couple of different aspects of actually using antibodies on the CosMx platform.
Yes.
both the morphology markers for the segmentation, and then a second question will be actually more broadly if you use the CosMx for broader protein panel analysis. So the first question on the segmentation with morphology markers, and I apologize if I didn't catch this. I was bouncing back and forth between doing a lot of stuff while you were presenting, but how end user tunable is that as far as, you know, if you go in and you have some difficult areas where your morphology markers might be a little bit squishy as far as, you know, a user's ability to quality control it visually and understand where different counts for different, you know, RNA targets or potentially protein targets are coming from and actually being assigned to for all of that subsequent, you know, elegant downstream, you know, spatial quasi single cell work.
Yeah. So the way the system works, obviously, it's a little bit different than GeoMx just because it's not a fluor conjugated primary antibody. It actually has a readout domain, and so a little bit different there. There is the ability to do some fine tuning, and primarily just like it does with the GeoMx, it comes into modifying the workflow a little bit to optimize binding. So there are things that we can do that'll optimize binding of that antibody. There's also ways that we can boost the signal a little bit, but when it comes down to identifying cell types, the beautiful thing about CosMx is you don't have to identify a specific, you know, region of interest or cell type. You can just lay down a pretty large field of view and then select the whole thing. And then utilizing, you know, spatial deconvolution, similar to what we've done with GeoMx, you can identify the cell type based off of the gene expression profile if you have some existing single cell data. And so we can certainly do that as well. So yeah, there's a lot of flexibility.
Yeah, and just to refine my question a little bit, because I might not have been clear. So some of the times, and it frankly, it's probably more of a problem with protein analysis than RNA analysis, or at least, you know, I haven't done any, you know, spatial resolve like Merfish type RNA analysis, but, you know, there can be issues where you end up, particularly with membrane associated protein markers. And so it's great that you do segmentation, you do cell type identification with whichever platform you have, but then you may be misassigning markers across cell boundaries, those types of things. And that was kind of what I was getting at, is how tunable is that aspect of the segmentation and the assignment or a quality control metric for those assignments.
Right. So the QC is pretty robust. It's not user tunable. We have an algorithm in place. If you feel that it's a little bit off, though, I wouldn't know of a scenario where we've seen that. It's just so new, I suppose. But yeah, we have the ability to modify a little bit, but really on the user end, it's a pretty set algorithm.
Cool.
Yeah.
And then if you don't mind, I'll switch to my second question, which is if we go down the road of the protein workflow for, you know, quantitating large numbers, multiplex numbers of proteins on the CosMx, again, just kind of from a standpoint of how do we control for the quality of our antibody staining on a run to run? If you guys with some of the preset panels have kind of come up with metrics for, you know, again, raw counts per antibody, those types of things, and just thinking about that, because that, you know, regardless of whether you're using a MIBI, you're using a CosMx, you're using any other type of multiplex protein thing, it's a heck of a lot harder to QC the performance on any given run of a single antibody versus the, you know, when we did it with 1 or 4 or 8 or something on a multiplex.
Right. Yeah. So similar to what we do with the GeoMx, we have a panel that includes proteins that aren't what you're profiling, but rather housekeeping proteins and then negative control proteins. So there'll be, you know, non-species controls. And so, yeah, those tend to work quite well. And that's really how we look for robust signal to noise and make sure that we're actually getting good counts. Yep. Yeah, you bet. Any other questions? All right. As things start to come up, please do feel free to reach out. I'm available. I'm in various cities at various times, but hopefully Fernanda and Grant can speak to the fact that I'm pretty receptive and pretty quick to respond. All right. Well, I hope everybody stays safe out there with all the snow coming down. It wasn't quite the snowpocalypse that we thought, and I've been shoveling. I can tell you it's pretty light and fluffy, so that's good.
Good to know. Use your legs, right?
Exactly. Yeah.
No, thank you, Jerid.
Thank you very
awesome
much.
presentation.
Yeah, you're very welcome.
Looking forward to continue working with you. I did drop in the chat. The next installment of the SIG meeting will be March 21st at 11 a.m. So we'll see a lot of you then. Thank you, Jerid.
Yep, you bet. Take care.
Thank you.